Hello students, today we will discuss ethylene signaling in plants. Ethylene is the simplest olefin gas and was the first gaseous molecule shown to function as a hormone. Ethylene is biosynthesized by plants and is well known to affect various developmental processes like seed germination, fruit ripening, senescence and abscission, as well as responses to various stresses like flooding, high salt, and soil compaction, etc. Once biosynthesized, ethylene diffuses throughout the plant and binds to the ethylene receptors to stimulate ethylene responses. Ethylene can also diffuse to surrounding plants and is the basis of the saying, one bad apple spoils the bunch. Means one bad apple could cause the others to spoil. Here, ethylene produced by an apple, means bad apple, hastens the ripening of others, right? Ethylene signaling components. First step in ethylene perception is the binding of ethylene to receptors. Plants contain multiple ethylene receptor isoforms. Early studies identified ethylene binding sites in the endoplasmic reticulum membrane, that is ER membrane, of plants, and subsequent research on specific receptor isoforms from various plants then confirmed that ethylene receptors are localized to the endoplasmic reticulum membrane. Now, in Arabidopsis, 5 isoforms have been identified and referred to as first is ethylene response 1, that is ETR1, ethylene response sensor 1, that is ERS1, ethylene response 2, ETR2, and ethylene response sensor 2, that is ERS2, and the last one is EIN4. Next is domain architecture of receptor isoforms. Different receptor isoforms in plants have similar domain architecture. Just look at this diagram. These 5 are receptor isoforms, right? ETR1 and ERS1 belong to subfamily 1, and ETR2, EIN4 and ERS2 belong to subfamily 2, right? Now, this is endoplasmic reticulum membrane, right? This is cytosol and endoplasmic reticulum lumen. Receptors are dimers. Just look at this diagram. Receptors are dimers. Dimer means a chemical compound made up of 2 similar or identical subunits or monomers, right? These dimers located in the endoplasmic reticulum membrane, right? Each dimer, one means this monomer and this monomer, right? They are stabilized by 2 disulfide bonds near N-terminus. Disulfide bonds, these small red in color, right? Shown here in this diagram. If you remember the structure of a domain having 2 ends, N-terminus and C-terminus. This is N-terminus part and this is C-terminus part. So 2 disulfide bonds are present near the N-terminus, right? So all the receptors contain transmembrane helices. Transmembrane helices, these rod-like gray structures, right? So you can see here all the receptors contain these transmembrane helices. Transmembrane helices comprise ethylene binding domain followed by GAF domain. GAF domain, this green in color, small structures, right? GAF. G means CG, MP specific phosphodiesterases, A adenylate cyclases, and F, FHLA, right? And kinase domain, right? This is kinase domain. Kinase domain, here the structure of ETR1, right? All this description is here is given is of ETR1. So ETR1 is a histidine kinase and other 4 isoforms are, that is ERS1, ETR2, EIN4, ERS2. All are serine threonine kinases, right? So here kinase domain of ETR1 having DHP domain, these yellow elongated structures, and catalytic domain, right? These purple colored structures. So histidine kinase having these 2 domains, DHP domain, elongated yellow colored structure, and catalytic domain, purple colored structure, right? So three of the five of these receptor isoforms contain a receiver domain, this pink in color, this structure, right? This is a receiver domain, and you can see here only ETR1, ETR2, and EIN4 having these receiver domains at C terminus of the protein, right? Now, in ETR1, this DHP domain of kinase is dimerizes, and a flexible region, this allows each kinase catalytic domain, this purple in color, to associate with this DHP domain, right? Now look at this structure. Receiver domains are predicted to be oriented away from the central axis of the receptor dimer, right? Now, signaling. Ethylene signaling may be canonical or non-canonical, right? Canonical, simple dictionary meaning acceptable procedure, right? So ethylene signaling involves ethylene receptors. These are ethylene receptors: ETR1, ERS1, ETR2, EIN4, and ERS2. Now we have discussed, right? So these are ethylene receptors. Then second, protein kinase, right? CTR1 and EIN2. These are protein kinase. What is the function of these protein kinase? They signal to the transcription factor which are present next to them, right? They signal to the transcription factor. Transcription factors EIN3, EIL1, and EIL2, right? And then two other transcription factors. These are ERF. They are other transcription factors, and ultimately leading to ethylene responses, right? So this is here ethylene, two ethylene receptors, two protein kinases, then transcription factors, then two other transcription factors, and ultimately ethylene responses. So this straight pathway is called canonical signaling pathway, right? Now, in this model, here this CTR1 is a negative regulator of signaling, right? Ethylene functions here as an inverse agonist, where it inhibits the receptors, which leads to lower activity of CTR1, releasing downstream components from inhibition by CTR1, right? Now, more recent evidence has shown that the existence of an alternative non-canonical pathway, right? Now, just look at this diagram. This is another pathway. You can see here, this is non-canonical pathway, right? In non-canonical pathway, ETR1 receptor signals to histidine containing AHPs, right? And then to ARRs to modulate responses to ethylene. So this is non-canonical signaling pathway, right? So this is all about ethylene signaling. If you have any question, please write in the comment section so that I can answer. So thanks for watching. If you liked this video, please make sure to subscribe for more. Thank you.